Getting started with our soil health webinar today. Again, I'm Holly Kirkendall, webinar coordinator for NRCS's East National Technology Support Center. I'm pleased to turn the webinar over to our moderator, David Lamb. David is the leader of the National Soil Health and Sustainability Team here at the East National Technology Support Center in Greensboro. David, you may now begin.
Thank you, Holly. Again, this is David Lamb, and I'm actually sitting in Ames, Iowa, and teaching a course that NRCS offers called Soil Health and Sustainability for Field Employees. And we're trying something new today. We're actually using Dr. Brinton's live webinar presentation as part of our course presentation. So not only am I here sitting in my room, I've also got 32 or 34 NRCS employees that are participating in course. So that's kind of exciting, being able to take advantage of this technology. Let me introduce Dr. Brinton. It's been my privilege to know Dr. Brinton, or Will, for the last 4 or 5 years since I've been involved with soil health. Dr. Brinton is a native of Pennsylvania, and he attended agricultural school in Chester County and Barnesville, Ohio. He managed a 20-acre organic vegetable farm for several years while he was attending agronomy school in Maine and Missouri. And he's also conducted graduate and postgraduate research on long-term soil health plots in Europe with a focus on soil biology. And for doing this, he was awarded a doctorate from the Unity College for his research on fatty acid respiration in soils and compost. And probably what most of you might know Dr. Brinton about, he is the inventor of the Slovita soil respiration test that we've used in our soil quality test, and it's very commercially available out there. Today's topic is the relevance of soil biology in assessing fertility and soil health. And with that, Dr. Brinton, I'll turn it over to you.
Okay, this is Will Brinton speaking. David, thank you very much for the introduction, and I want to also thank you for the invitation and the opportunity to address this audience today. This is a very exciting topic for me, and I should say right at the get-go that no one addressing such a range of interest here can profess completeness. It's a very, very broad field. Today I've chosen a particular emphasis, and it's really based on personal preference. 6 months from now, I may be developing another arena of work in this exciting opening field, and my addresses would appropriately change. I did select the word relevance in this title, the relevance of soil biology. It's an old story, but a long time ago as a youth in a land-grant college, I was told that soil biology was irrelevant for crop production. And when I suggested it in a syllabus, I was told only somebody with a vivid imagination would suggest such a topic. So we've come a long way, and for many of us, it's been many years of quiet development and a slow learning process. Let me take the first slide here. So, my theme today concerns the potential and, in fact, the necessity of including soil biology in routine soil testing. As this image suggests, we have a situation today where farmers are very, very familiar with soil testing, and the most commonly reported parameters in soil testing are phosphorus, potassium, and pH. We are addressing the topic as to whether additional parameters of soil biology will be relevant in this discussion. Last year, I surveyed a number of soil commercial labs and discovered, according to the survey results, that nearly half of the labs have at some point considered some form of nitrogen mineralization potential test as a means to attempt to adjust fertilizer nitrogen recommendations. However, the majority of labs still only use nitrogen replacement tables, expected yield tables, and so on to decide these nutrient rates. Many people today can jump into the soil health arena at different points on the circle. For me, commercial soil testing with its broad reach and force in mainstream farming is a suitable and very challenging starting point. In a way, my focus today is a business-to-business model where we're reaching and working with soil labs around the world to help bring cost-effective testing into the discussion so that farmers have available new pieces of information that they didn't have previously. Okay. Let's find my pointer here. I wanted to start with this particular slide of yield trends over the last 150 years. We certainly stand at an interesting junction in time today. We all look back to astonishing yield growth in the last 60 years, particularly in the case of corn, using that as the prime example. After World War II, inexpensive nitrogen became widely available to growers, and the race was really on. Often overlooked here in attributing yield gains to fertilizers, however, are the heroic selective breeding efforts that have been focused largely on corn, but of course in the case of many other crops. These have played a significant role in these yield increases. In fact, on this particular diagram, one researcher recently suggested that this yield trend here might have gone more like this had it not been for all the breeding efforts with these crops. Now, I want to draw your attention to this graph in a different sense. I want you to look at the last 100 years before inexpensive fertilizers became available and look at the yield trend. Now, I'm just going to draw a line across here for the average corn yield, and notice for nearly 100 years we had steady, sustainable yields without varietal improvement and with very minimal input of fertilizers. What explains that? Whereas today we look back at those as pitiably low yields. They were sustainable yields, and farmers were living off the bounty of nature. They were enjoying the benefits of rich soils, very healthy soils, and especially high reserves of organic nitrogen that kept these soils alive and going. And what we're trying to do is draw your attention to this potential of soil to have a living organic reserve, and finding ways and methods and testing devices to begin to include that in our fertilizer planning process. Now to some of the startling news. We often applaud ourselves for these yield gains that I have mentioned, but there is emerging concerns on all fronts. A recent published study in Nature, I show the reference on the far right of the screen, examined world crop trends and provided clear evidence of stagnation of yield growth for all major crop regions and major crop types around the world. According to these data, it is evident that yields of major food crops are no longer necessarily increasing. Not only this, the study also examined areas of the world where yields have never improved, and these include all the major hemispheres and major food categories from corn to rice, wheat, and soybeans. Now, the study also reported that there are still regions in the world that are experiencing yield growth, but it pointed out, and counterbalancing this, the study also found evidence of yield collapse in all major crop categories. This is where forces build up and cause a decline that appears to be not fully controllable. Clearly, we need to find the missing ingredient here, which lies beyond chemistry and breeding, I believe. And my hypothesis is that this is soil biology. So, how did the study attribute these trends? This is all right out of the published paper. The authors emphasize that there are a number of causes for yield stagnation, including climate change stress, which is well known, salinization, soil erosion, pest and disease buildup, and finally, depletion of soil fertility. A key question here is, are we digging deep enough to understand what depletion of soil fertility really means? It could just mean imbalance of nutrients. It could just mean lack of potassium, but the authors pointed to some of the deeper ramifications of depletion in terms of the overall resource that constitutes soil fertility. If the only answer is better nutrient management, then perhaps the future is simple. more and better applications and more careful technology to tweak the system. But if we are dealing with biological factors as interactions here, then these bets are not as certain. And this is our topic today, and there is increasing urgency to it. So you're all familiar with the theory of collapse based on the model of the three-legged stool. So the way it works is take any of the legs, which I've called biology, chemistry, and physical factors, and knock one out, and the whole system collapses. It works the same from any vantage point, with erosion standing in for loss of the physical, or salinization and nutrient balance for loss of the chemistry leg. Any one of these in an appropriately damaged fashion will bring the whole system down. So it seems foolish to tempt fate and abuse the leg of this, any leg of the stool. However, we are dealing with a much more complex symbol than a three-legged stool. In fact, you could call it 5 or easily 6 legs for farming, and you get the picture. In actual fact, there are so many interacting factors, as this diagram indicates. There are so many individuals, farmers, and agricultural industries working on individual aspects that the likelihood of experiencing any collapse seems very remote. But I observe among growers, in fact, and agricultural professionals, is a fair degree of confidence in the system. But the fact that this complexity is there should be no excuse to lose sight of any one of the individual legs, and especially what I believe is the most overlooked one, the leg of soil biology and soil health. How do we come to recognize and measure these important and concealed factors and learn what the actual associated role is within such a complex environment? Attributing factors like this requires complex analysis, multivariate analysis, the careful removal of factors to make other factors become evident, and all this is very challenging work. As I indicated earlier in this presentation, early farmers in America enjoyed stable yields by living off biological capital, and yet they weren't aware at the time. When the principles of Liebig and Mitscherlich were laid down in Germany in the late 19th century, there was no concept of soil biology at all, and that was the beginning of modern agricultural science. I want to address here a little bit what I call the biological underpinnings of yield. I've already indicated this in the earlier slide where I suggested that 100 years of stable yields in early America was the gift of nature and the gift of rich soil. Here we have a study that Rick Haney and I tabulated data from, where instead of taking the yields and the soil quality from plots with fertilizer, we excluded all the nitrogen plots and took only the control plots for yield. Here you see on the y-axis the corn yield per bushel, and on the x-axis we used our 24-hour CO2 burst respiration test. What that test is, and I will describe it later, is one way of measuring the soil's biological capital. But look at the astonishing correlation here between the rate of respiration, which follows this axis here, and the level of yield without the nitrogen. Now, we often refer to areas of the country as having higher or lower background yield potential, and we're trained in agronomy to factor this out by subtracting out these plots when we do research. But in fact, these plots, like the placebo effect almost, are very, very important to us because it provides evidence for the biological capital present in soil, which in this one region here might be a low yield potential, but here's a region of the country where control plots are yielding 90 bushels of corn without any nitrogen. What we're basically saying is that's nature's process. The biological reserve in the soil is carrying these yields. Now, if you did a fertilizer rate study here and ignored the nitrogen potential in the soil, you might suggest, in this case, everybody gets 100 pounds of nitrogen. In this case here, it would make up the deficit very well, and here it would be an extreme over-fertilization. These are the kinds of concerns we have nowadays in terms of where we are going with increasingly expensive fertilizers, nitrogen that's found frequently in surface water and groundwater, and the concerns of the agricultural economy and the profitability of farming. So let me address another angle of testing that's very interesting here. And this has to do with, for those of you who are familiar with PSNT, pre-sidrous nitrate test, a method that's been around for at least 20 years and has been found to work very well as a predictive tool for whether a response to nitrogen would be anticipated or not. I'm concerned with soils that perform well when the soil test data predicted more nitrogen need, and I've already indicated that in this upper quadrant here. This resulted from an interesting discussion we had a few years ago at a Rutgers University conference where we addressed the anomalies in soil testing that biology may explain. How do you explain the fact that a soil early in spring has inadequate levels of nitrate in the soil and yet goes on to yield between 80 and 100% regardless of additional fertilizer? The explanation may be the biology itself. These are soils which, had we tested them for biological properties or respiration or nitrogen mineralization potential, we would have most likely discovered they had a very good potential, but factors such as a cold spring or a heavy rainfall in the spring leaching nitrate out gave the test a false negative result, indicating a farmer should be applying nitrogen when in fact he did not need to. So what we are trying to do is not replace all these test methods. What we are trying to do is add in new testing approaches so that we can account for these factors. Now, had we done a test on these soils down in this range here and found very low biological potential, and you found also by PSNT low nitrate, you would definitely have evidence there that that soil would require more nitrogen to reach a satisfactory yield. Now let's stand back a moment and look at the biological picture. My focus is the whole functioning of the soil system as expressed by overall respiration. Respiration is the metabolic exchange of oxygen for carbon dioxide, which is common to all living organisms. These charts provide a generalized breakout of the soil system, beginning with organic matter on this side. If you break down organic matter, 85% of it in soil is probably humus, 10% is plant matter from roots, and 5%, seemingly relatively small amount, is this fraction of living soil organisms. If you then take that fraction of 5% and break it down here, you have these categories of algae and fungi, bacteria, actinomycetes, soil animals, which include arthropods, earthworms, and so on. Roughly 50-50 bacteria and algae, and a slightly lesser quantity of soil earthworms. Some of my early work focused on correlating the population in the soil of animals versus respiration, and I will show you some of these results later. It turns out that this relatively small slice here of living organic matter, the biology of the soil, is doing a tremendous amount of work for you. It seems small, and if you look at the level of organic matter in soils today that's down to 2%, in some cases under 2%, 5% of that is a diminishingly small number, yet these microbes are performing vital and quantitatively measurable functions for the soil system. Let me try to quantify it here using an example of a soil with 2.5% organic matter. Immediately, it leads to an astonishing number. In top 6 inches of soil, 2.5% organic matter suddenly translates into 50,000 pounds per acre of organic matter. If the organic matter is at steady state, there is 1/10 of that present as organic nitrogen. In other words, 5,000 pounds of organic nitrogen could be associated with that. What is very interesting is that living off of this is that 5% range of microbes, so you have 2,000 to maybe 2,500 pounds per acre of microbial biomass living off this organic matter. Now, many people just focus on measuring this factor, or microbial biomass used to be a test that was popular and is no longer so much the case today because we've moved more to the activity of it. What are the microbes doing? And the first obvious thing here is that they are kicking out all this carbon dioxide. And we are beginning to quantify it on a daily basis, and the numbers are somewhat astonishing. Did you know that soil in this category, under normal, moist, warm conditions, may be emitting 100 pounds per day of carbon dioxide per acre? Now, that alone may seem just an academic point, but look at what's associated with it, and these are two very important factors. First of all, that carbon dioxide roughly matches the amount of CO2 a plant, a canopy of plants at full growth, has to assimilate on a daily basis to maintain its dry matter accumulation. Isn't it a coincidence, or is it that the soil, under normal conditions, is actually producing the carbon dioxide that the plant needs quantitatively to maintain its growth? At the same time, if you measure this, the turnover of CO2 every day is dumping 1 to 5 pounds of nutrients per day into the system. When you look at it this way, it seems somewhat surprising that in fertilizer studies, in fertilizer theory, there is no concept of the soil generating the nutrient on a seasonal basis, let alone on a daily basis. We are able to validate that under these healthy conditions, these soils are capable of moving this quantity of material, both carbon and nutrients, through the whole system. And this is what we're trying to do. We want to find ways to measure it more accurately so that we can have more meaningful results from testing these soils. Okay, let's stand back now and look at the whole system. If you study soil biological factors in depth, you begin to see how traditional nutrient theory vastly oversimplifies the system. It's been convenient to us for the last 150 years, but I think increasingly if we continue to ignore the other dimensions, there'll be a big price to pay for it. The whole system is comprised of moving parts, and when I refer to soil health, I think it's a misnomer to just say it's soil health. It's the whole system that we're talking about that makes soil healthy. You can't just look at the soil alone. It's the system with the living plants in place and the exchange with the atmosphere that creates it. With my focus on respiration of soil systems, let me point out to you what I call the CO2 transactions are actually performing on a daily basis or seasonal basis, whichever way you want to look at it. Number one, carbon dioxide transactions are associated with furnishing soil animals with food. Number two, they are associated with sequestering CO2, an increasingly hot topic about where is all the carbon dioxide ending up. Thirdly, carbon dioxide released from soil during normal metabolism is meeting the plant needs for carbon dioxide by means of photosynthesis, a very, very important factor that we have known for nearly 100 years and simply chosen to ignore, mostly because we seem to think it takes care of itself. Another factor is that this exchange of carbon dioxide is directly associated with releasing available nitrogen and phosphorus and other elements. Nitrogen certainly the chief ingredient, since 10% of organic carbon in the soil is probably present as nitrogen with very little variation on that, and about a fifth of that as organic phosphorus. And finally, and another astonishing fact that we tend to overlook, carbon dioxide dissolves in the soil solution in the natural water falling from the sky and forms weak acids called carbonic acid. What does that do? It dissolves soil minerals. It's a natural weathering agent. So the soil has a whole system set up to move and the nutrients, move the carbon and the moisture through the system. And it comes down to what I like to say, that nature is balancing several equations simultaneously. Let's take a little bit further, more in-depth look at this. And I have, in the last year and a half, been stressing to people that we have to look at the respiration of soil in a broader light. And I've drawn on work I did in Sweden many years ago, where I traced the lineage of respiration testing in soils back to an early figure named Lundegård, a soil scientist who did all the early development work that found its way all the way to Rutgers University between the wars, which validated these early assertions, and which are very astonishing. Did you know that crops are very hungry for CO2, requiring 100 times more of it than nitrogen on a daily basis? In fact, Lundegård quantified these relationships, and I have revalidated that, and anybody can do this themselves just by multiplying the dry matter yield by the carbon content and determining what your crops are actually sequestering. But wheat requires, in the absence of any soil respiration, wheat would need to filter 10 cubic acres of air per crop acre per day to acquire 110 pounds of CO2, which it needs for plant assimilation. Corn is even more staggering in this regard. 38 cubic acres of air would be required per day to be filtered per acre to provide that crop with 410 pounds of CO2, which it needs to complete photosynthesis. We are beginning to hear reports these days of carbon dioxide limitation in crops, and against the backdrop of concerns about increasing CO2 in the atmosphere, there's a tremendous amount of interest but also confusion on this topic. There's another way to look at it, and this is one way that I like to present it that comes from studies that we are doing here at Woods End Laboratories. Where is all the CO2? If you ask the average person in the country, they'll probably say, oh, it's in the atmosphere and it's rising. But in fact, in the atmosphere, we have only 400 parts per million, if even that, in CO2. And as you approach the soil, if you come down towards the soil, particularly a healthy soil with a crop growing on it, and you measure CO2 as you approach the earth, the levels start increasing dramatically. When you come down into the plant canopy, you can see levels as high as 800. When you come down here into what we might call the thatch or litter layer on the soil, as much as 1,500, and finally dive down into the soil pores and measure carbon dioxide, you will see levels as high as 4,000 parts per million. That's 10 times over atmospheric levels present just 2 feet above the soil. It might be useful here to ask, what is nature doing? I tell students to take these values and convert them using the ideal gas law into moles of carbon dioxide and then compare it to the soil and plant carbon content. And you suddenly begin to see the method in all this complexity. It's easy to see what I meant, that Lundegård, way back in 1927, discovered really that essentially nature is feeding plants their CO2 from the soil to maintain the photosynthesis while plants are growing. In other words, this gives us a new angle in understanding the preeminence of soil health from a holistic angle that's rooted in excellent science. We want to get to the basis of understanding this. Now, I don't need to say here how different this is in a depleted soil environment. If you have a depleted system, you can be dealing with only fractions of these levels present in the soil. So let's just jump down into the soil and look at how dynamic this is. I took this picture at a Pennsylvania No-Till Alliance meeting last summer at Fertility Farms in central Pennsylvania, and it provides stunning evidence of this dynamic interaction of earthworm canals, roots pursuing the canals, and enrichment of soil. It is well known from numerous published papers that worms transport organic matter down into soil layers. They provide aeration, and water channels into the soil. We measured this level here was about 28 inches deep in this example. Published research also shows that if you analyze these earthworm canals, if you cut the soil out and look at the area around where the worms have passed, they are enriched with carbon, very high in nitrate, and very high in available phosphate. In other words, the worm itself is transforming the soil as it passes through it. The other most interesting thing is worms stimulate microbial CO2 rates. Whether it's a direct or an indirect effect, this has been substantiated in a number of studies and was the basis of my early graduate studies in Europe, but we wanted to find the association of these factors. It shouldn't surprise, if you look at what healthy roots are doing in soil, it shouldn't surprise you that they are associated with significantly increased soil animal population, bacterial population, and CO2 respiration. This is a recently published Canadian study where they had quantified root mass under stress conditions, under moderate growth and vigorous healthy conditions. But look at the change if they took the square feet of root area in these different scenarios. So it's an exponential increase from a poor soil to a vigorously healthy soil in terms of the amount of footage. If you laid it out on a surface on a single dimensional plane, each cubic foot of soil had up to 852 square feet of rootlets available for respiration and organism feeding, as it were. In my early work, we focused on correlating soil CO2 respiration with earthworm counts. And the way we did that, we measured earthworm canals in the soil by digging down 10 centimeters, scraping the soil off, and counting the canals present. In this particular study, it resulted from a 17-year, a long-term field study, 17 years of different crop rotation treatments that were tested. I learned to do the 7-day respiration method, which is on the right. This is this range of values here. And the 1-day method on the left. And both of them, notice the sharp correlation with the earthworm canals in the soil. Incidentally, in these particular examples, here's the control plots right here after 17 years of treatment. Control plots had no fertilizers, no improved management. These three variants here, in this particular assay and this, are the inorganic fertilizer plots. No cover cropping, no intercrops, just inorganic nutrients. They were even lower in respiration, and particularly in earthworm canals, than the other ones. And what were these? These were animal-based farming systems, all included manure in the rotation. So we already knew back in the late 70s that there was dramatic evidence that improved crop rotations and bringing animals into the system had a dramatic effect both on respiration and the population of soil animals. I'm going to take the opportunity right now to pause in case there are any questions. In the final part of my presentation, I'm going to focus on actual soil testing and go into the work that we've done with soil respiration and organic nitrogen analysis. David, if you are present, you could indicate that there are questions. Otherwise, we will continue.
Well, there are some questions there, Dr. Brent. I guess the whole concept of CO2 and plant growth, so, again, I don't mean to sound stupid here, but can you talk a little bit more about filtering, or that slide you showed where the cubic feet of or acres of CO2 to be removed? Is that something we actually would have to filter through the plant, or the plant would have to take in through its above-ground respiration? Is that what you were saying?
Yeah, absolutely. The plant has to acquire the carbon dioxide. Now, the carbon dioxide is transported to the plant, and the early studies tried to pinpoint the physics of it. What was the mechanism that it got? And they mainly found that it was convection forces that carry CO2 to plants. The mass diffusion just wouldn't work with such a low amount of CO2 in the atmosphere, only 400 parts per million. The idea that plants would be capable of obtaining CO2 that would diffuse down from as much much as a mile above the ground to the plant was ruled out. What was actually found was, wind movement and convection forces moving carbon dioxide across the landscape is the primary mechanism of delivering a stream of CO2. But as reports are beginning to evidence, when you have a very dense canopy, particularly a very dense canopy of wheat or corn, and it's already been shown even with sugar beets, the carbon dioxide demand of the plant way exceeds what the convection forces in the air are able to deliver. But this is where the soil comes in. A healthy soil is capable of delivering that amount of carbon dioxide to the plants.
So in essence, the soil is providing the CO2, and it's not being removed from the atmosphere.
The soil is fundamentally applying the CO2 to plant growth. That's nature's plan. Now, could we split hairs and try to determine which it is? It would be some of the both. It's obviously going to be CO2 in the air, and what CO2 emits from the soil that plants do not take up will end up in the atmosphere, but plants will cycle it back down into the humus.
Okay. You talked about respiration and measuring it. Is there a magical number of respiration that you need to be above to get the benefits that you've been kind of talking about?
A maximum amount? Well,
no, a minimum amount. You need to achieve a 30 or 40 or whatever rating.
Yeah, we are working now carefully, and we're developing a field CO2 test kit kind of approach where a farmer could go out and get some idea what is the CO2 production capacity of the soil at any given time. Now, you can imagine the complexity of attempting this kind of measurement because it's so dependent on temperature and moisture content in the soil. The work that I've done with Dr. Haney at the ARS lab in Texas, our goal was to create a laboratory method that soil laboratories could implement that represents this CO2 potential. Nevertheless, it still remains a question, how is it functioning in the field itself? So we are working on producing some tables now that show the amount of CO2 produced per acre and the amount of carbon dioxide that your crop would need, which obviously depends on what stage of growth it is at. But this can all be quantified. And I've seen some interest from crop consultants in the Midwest to try to do some canopy studies this coming year to see if we can get a better handle on this.
Okay. Now, let me ask one last question. I'll let you move on. The whole issue of earthworm holes and movement of phosphorus, dissolved reactive phosphorus down through these holes, and then you're talking about CO2 being increased by the earthworm activity. Can you kind of talk about that a little bit? Is that a good thing, a bad thing? It just seems like we're getting a lot of mixed messages about earthworms out here.
Well, I tried to indicate that this is not necessarily a cause and effect relationship. As I indicated previously, if you look through the literature, there's quite a bit of evidence that earthworms are associated with stimulation of overall respiration in the soil. One thing is all the bacteria that are passing through their gut are being fed at a very high rate and have much higher respiration. There may also be secretions and so on associated with passing through the soil that stimulate microbes. But flip it around, flip the cause and effect around. Earthworms are present because of the practices you are doing that bring living organic matter into the soil that they can feed on. So I don't want to divorce it from the reality. If you just look at the screen image and the plant layer that I show there going down into the soil, that's the food that the worms, bacteria, fungi, and other soil animals are seeking. So you've got to see it in context.
Okay. Why don't you continue on, and we'll have some time at the end of the presentation for a few more questions.
Okay. Thank you very much. Now, we're going to go forward and dive down into this area of respiration testing, which is very dear to my heart, and I've spent the last 38 years focused on different techniques, technologies, and methods to do it. The big complaint, and again, I'm focused really on business-to-business relations with other professional soil laboratories. The big and the valid complaint is we have so many technologies out there capable of doing this, some of them very, very precisely, but it's not cost-effective in a soil lab. It's not cost-effective to offer it as a service. And at one point, I felt frustrated, and I said to a laboratory, I am so tired of hearing this excuse. It's a valid excuse, but what if I make it more cost-effective to do the testing? Will you ignore it then? And they said, I suppose we'll do it. So we set out, and I have this picture here just to show this huge array of equipment. I have all this up and operating in my laboratory at any one given time. We can choose any type of method. For example, down here we can put soil in this unit here and monitor its respiration over a 120-day period. It has about 18 channels on it. Would you be able to offer this commercially? Not without receiving a great deal of money. A fast method that Rick Haney and I have used quite a bit is using these infrared analyzers here. Again, you're limited by the amount of jars here that you can test, and you actually have to break the system down and refresh it every day because of the interference of moisture on the infrared detectors. These are also very costly machines. I will hardly mention base titration. This is a method we all grew up in, and I was a willing student once sent down to the basement to titrate CO2 because nobody else wanted to do it. It's time-consuming, and you have to standardize your reagents, and there's no way you could bring that into a commercial laboratory setting. In the 60s, gas chromatography emerged. That's what you see here. And with GC, you were able to identify different gases in the soil, as we're doing right here. This one actually is separating out oxygen, nitrogen, and carbon dioxide, and methane even, as it comes out of the soil system. Again, this is the kind of technology that is barely cost-effective in a production laboratory. Each one of these runs takes over 30 minutes. I still use an old-fashioned manometer that I found was built around 1860. The early European scientists that discovered and worked with soil CO2 did have very precise technology, and it was all measuring the CO2 by volume method, as this device here does. And they were able to get satisfactorily accurate readings a long time ago. And finally, flux chamber work has come back in fashion. A flux chamber, you place it over the soil surface, and it fulfills an air exchange in a studied fashion so that you can assert that you are capturing CO2 as it accumulates, but not allowing it to overaccumulate in the system. This kind of work is being done for CO2 climate change studies quite a bit now. Again, none of these methods were popular in soil labs, and so soil biology has been overlooked. What we did is turn it into a mission, and this was part of the reason I signed a CRADA agreement, a Cooperative Research and Development Agreement with Dr. Haney's laboratory and that ARS about 10 years ago. We wanted to see if it was possible to develop methods that could be taken into a modern laboratory environment. And what we did was we created a way to capture these gases in these what I call probes here. These have reacted with carbon dioxide. When they're produced in the factory, they're standardized blue, and when they react with the soil, they turn different shades of green and yellow. And you can run hundreds of samples a day with this system. And with that in mind, we were only at the beginning of their mission, which was now to take this to soil labs all over the country. I want to kind of dive in here. Here's the method as Rick Haney and I developed it, and we called it CO2 burst after a phenomenon that was first observed both in Europe and Africa in the 1950s, where soils subjected to drying and then sudden re-wetting, either from irrigation or rainfall, would experience a burst. And this burst here is shown in this diagram right over here. When soil dries, microbial activity drops to almost nothing. And when you have a rainfall event, it increases. It doesn't increase and just stop at baseline. The dotted line here would be the baseline. You experience a pulse or a burst, defined as here, and this is what we decided to measure. It's an indication of the entire potential that was missing while the soil was dry. If the soil is not dried adequately, as in this example here, and it's remoistened, it comes right back up to its basal condition and continues respiring. In other words, there is no pulse. So we had to design the method to control these parameters. Here's what the test reveals when you look at different soils. These are samples that actually Ray Archuleta collected for us a couple years ago and sent from North Carolina. I want to start with this fairly abused soil here at the bottom, continuous truck farm, high tillage system, respiration 12 parts per million. Very, very low. We can measure down to 3 parts per million, but that's so tiny there would not even be any nutrient exchange associated with it. When we looked at some cover crop farms in the area, we had levels 3 and 4 times that. In fact, here's a multi-species cover crop farm that on similar soil types had attained almost 7 times more respiration than this continuous tilled soil. This was a bare tobacco farm soil. There was enough litter, I guess, entering the soil to have a somewhat reasonable respiration. But the most interesting one for me was studying a no-till farm in the area where even the subsoil of the no-till farm had significantly more respiration than the topsoil of a neighboring tobacco farm. And the topsoil was fairly decent. We have since gone back and remeasured this farm, and they're in the 70 part per million range. But look at this example here in the bottom right. This is Virginia prairie soil from a farm in Nebraska that was sent along with the shipment. This is the kind of value we see in rich soils, between 70 to well over 100 parts per million per day of carbon dioxide. So these results, when they're related to the past management practice, give you a unique perspective on soil management. Now I want to come to a somewhat alarming, if not surprising, discovery we've had very recently in trying to learn what are the forces in soil that are challenging a respiration test and causing unusual variability. In the wetting method that we use that Haney and Haney published, beakers are placed in water and the water moves up by capillary action as shown here in this left picture. This is a well-structured soil which after re-wetting, if you take a soil spatula to it, you can see it has very nice crumb structure and porosity. But we have now been encountering soils where with the same type of method that we have developed, they immediately oversaturate. And you take a spatula to the soil and it's undoubtedly at or exceeding 100% saturation from normal capillary methods. So we have been working in the last year to pinpoint this because this phenomena here negatively affects respiration. And it does so very variably, so it's been frustrating for labs in some occasions reporting differing results on the same soil. Work that I've done with Bruce Hoskins in the University of Maine lab have shown that the same soil sampled differently and ground differently can suddenly go into this overwetting state, and then the respiration will plummet. We have identified these kinds of soils as structurally damaged soils, and we have also found a link to soil laboratory management, and that overgrinding of soil samples disrupts and destroys the particular structure that protects and preserves porosity in soil, and enabling CO2 aeration to be aerobic. This particular soil here was a proficiency sample soil. Proficiency soils are sent out in America to help soil laboratories gauge their capabilities to extract and accurately measure chemical nutrients. We have been using these in our biology program, and all these soils re-wet fairly spontaneously using the method that we have adopted so far, because they have been ground to pass down to 0.7 and 0.5 millimeter sieves, which improves the veritability and reliability of chemical extractions. So in this work, what I'm trying to indicate, we have now encountered a new challenge that maybe we need a different way of preparing soils if we are going to have biological tests represented in proficiency programs. And this is an area that I am focusing quite a bit of work on right now, because many of the laboratories that we work with depend on these programs for their reporting standards. I want to also mention an exciting new procedure that we developed called Labile Soil Humus Amino N Test. 10 years ago, actually in the 1990s, our company was hired by USDA Technical Center in Chester, Pennsylvania, to examine ammonia losses from manures during routine animal-based farming. And we had found that when manure is stacked in composting scenarios and allowed to get hot, it was losing huge amounts of nitrogen, as much as 85% in 120 days. And during that project, I developed a chemistry probe to detect the ammonia on site because we had difficulty transporting samples back to our laboratory, measuring them, and reporting the results. It was 10 years ago while Rick Haney and I were discussing how to represent or get at the organic nitrogen component of soil. He actually, just out of the blue, said to me, "Will, you have an ammonia probe, why not stick it in soil?" Of course, at first I thought it wouldn't work, but it did work as long as we released the ammonia factor in the soil by a suitable reagent. We've had some reports recently. Carl Grillard at UConn has published a paper showing a very high responsiveness of our SLAN readings. In other words, we're now indicating the organic nitrogen that's being set free when you treat a soil. This is nitrogen associated lightly with humus. That is relatively available. And we're getting very good predictability between the range of 100 and 200 parts per million in soil. I'm happy to say that I will be working with IS&T methods and Dr. Mulvaney's laboratory to see if we can recapture some of the correlations shown with these other methods that were developed to reveal humic nitrogen compounds. But if this works out well, now we can cover both the respiration and the accumulation of organic nitrogen reserves that is the hallmark of healthy farming methods. What do these probes look like? It's even a more interesting picture. Those are the same 8 soils that we looked at. And take a look at this. This is Ray Steyer's farm in North Carolina. It's been no-till for at least 30 years. If there's no organic nitrogen present, as there is in this continuously tilled soil here, we get very low readings. The lower limit of detection is about 50 parts per million. The soil is completely depleted of organic nitrogen reserves. And look at this no-till soil here in the middle. It's as high, actually it is higher than virgin prairie soil in Nebraska. We know that these levels of organic nitrogen are associated with very decent yields. In fact, I will show you a slide later that Ray Steyer's farm has achieved almost nitrogen independence in recent years. A good example of this, if you visit the old rotation plots at Alabama Ag Experiment Station, are these long-term cotton plots. On the left, there's continuous cotton with no winter legume, and the plants are very yield-stressed, requiring careful nutrient supplementation. Here on the right, just the presence of a winter legume is enough to make an enormous difference on these crops. And what I am saying is that these soils are accumulating organic nitrogen that we now can measure and begin to use to account for the sources of nitrogen and how to protect it in our soil testing programs. Here's a picture of Ray Steyer, no-till, zero inorganic nitrogen corn. His results, whenever we test the soils, he has medium-high CO2 respiration and very high soil labile amino nitrogen in his results. In other words, our tests predict this performance, and it matches what he is finding. Overall, I guess I'm trying to summarize here ways of reporting results of all this kind of testing. We can use it as an index of soil biological activity. You don't necessarily have to quantify it, but look at it in terms of low, medium, and high, and help a farmer gain a sense if they're doing the right thing. Clearly, that leads to monitoring soil improvement. Are we treating our soils in ways that are increasing the CO2 factor in the soil, increasing the accumulation of organic nitrogen in the soil? We can compare it with strip plot results. I often tell farmers, just split the plot and put down your regular rate of fertilizer here and use a rate on the other side that would be indicated by our kind of testing. These methods can be integrated into soil health lab tests. just about any lab performing soil testing can find a way to integrate them. We certainly are able to indicate potential nitrogen mineralization. There are many studies underway now. We're learning a lot about the reliability of testing and the factors in soil that make it a better or worse predictor in terms of respiration and other factors. And finally, with this land test, we see the potential to have an indicator of responsive and non-responsive soils. Here's just an example of interpretation that we use now. When we work with this amino nitrogen test, we're able now to categorize soils into three groups of very likely response to nitrogen, maybe response to nitrogen, and very unlikely response to nitrogen based on the levels that we observe here. We then compare it to levels of respiration, either by the Haney-Brinton CO2 burst test that's shown here, or if you do basal respiration in the field, the numbers are smaller because the conditions are different in the field than in the laboratory. This helps you get a sense of how to incorporate this into soil testing. So there's a lot of interest in soil laboratories in these methods, and I could show a lot more pictures here of different and new forms of soil health tool results that are available. Presently in America, about 5 laboratories have adopted the soil health tool that Rick Haney has developed, which incorporates the Solvita respiration and other parameters. In our test, which is shown here on the left, we have a box here showing indicators, and we do include aggregate stability testing in our ranking and include it in our overall score. Ward Laboratories, A&L Biologicals, several of the A&L labs are now offering this, Midwest Labs, Brookside Labs, and others. So farmers now are gaining access to this kind of test result and needn't say that they can't get it. In the middle here, I show a report actually from the United Kingdom. This is NRM Labs is a soil lab that we have been working with, and they have come out this year with the soil health report. It incorporates in the center part of it a whole biological rating scheme, and then it compares the ranking to the soil texture and the erodibility of the soil. Finally, as a summary of this presentation, I want to point out that nutrient-based yield theory has served us well for 150 years. But don't we need to change this? We need, in my opinion, to develop a biological corollary for nutrient yield models. Across America, farmers, crop consultants, and soil labs are testing new tools, and this is very exciting for us. There's a tremendous amount of business-to-business interaction. We've had exciting seminars with crop consultants and soil scientists working in the field to help bring these methods to the public. Finally, I'm concerned about the future of yield stability, as are all growers. But I believe that soil biology is the new frontier to stabilize yield growth. And I think this is something that we can all see a tremendous amount of work and development on. I want to take the opportunity to thank you all for your participation today. I've provided some information here for contacting me. If you wish, I would love to hear from you. And if there are further questions in the future, I will be very happy to respond to them. David, I will turn it back over to you for you to ask any further points.
Thank you, Will. That most excellent presentation, and from the comments and questions I got, I think our audience would agree with that. And there's too many questions to just pick out one or two, but how would you see what you just described being applied on a 1,000-acre farm? Could you describe, how biological testing would be implemented on a big-scale farm like that, or what could you envision?
Well, we are doing soil testing on many very large-scale farms. And basically, we interact with the crop consultants in the labs working with those farms to bring our program into the existing program with very little changes. You can actually use your routine soil testing process to obtain this kind of additional information, and our goal is that it should not cost significantly more to do that, it should be readily available to the growers. Now, having said that, can you do, for example, soil health testing in precision farming? I don't see why not. It's just a matter of starting to do it and mapping it. I see precision grid mapping of farms based on available nitrogen, phosphorus, and so on. Why not add soil health to that? What if, when you draw all the connecting lines on those maps, you can see a different perspective across a large field than shown to you by available nitrate or so on? If farmers are only doing precision fertilization based on available nutrient mapping, they may be overlooking very important underlying factors that we can add to it with these streamlined tests that modern soil labs can perform. So I haven't seen any farmers, including some, we've sampled farms that are 2,000 acres in size, that are expressing any significant concerns about the availability of this technology. Certainly the implications are enormous in terms of potential fertilizer savings, and that simply goes to the bottom line for the growers.
Okay, and I think that's probably a good way to end this, and again, thank you for this excellent presentation, and those who want to, I know I'm going to watch it again. There's a lot to gain from it, and it'll be available for replay off of the conservationwebinars.net site, as Holly mentioned earlier, tomorrow. And with that, I'm going to end it and turn it back over to Holly. And thank you, Dr. Brinton.
Thank you very much.
Yeah, and let me express my thanks to both Will and David for making the presentation and moderating today. We had more than 360 people join today's webinar. Participants, if you selected to earn CEUs for the webinar, please return to your open browser window to continue the process that's offered by Step 2 at conservationwebinars.net. And with that, this concludes our webinar presentation today.